welcome. If you are settling in for the evening, finding your position, letting the quiet gather around you, letting the thoughts of the day begin to soften at their edges, then you have come to exactly the right place. This is the Science Channel for Sleep, and tonight we are exploring something that is, in every meaningful sense, the most extraordinary object in the known universe. It is not a distant galaxy or a black hole or a nebula of swirling gas and light. It is closer than any of those things. It is, in fact, inside your head right now, processing these very words, generating the experience of hearing them, and, if all goes well tonight, gently guiding you towards sleep. Tonight, we are exploring how the human brain works. Before we begin, if you enjoy this kind of calm, unhurried journey through the wonders of science, if this is the sort of content you like to fall asleep to or rest quietly alongside on a still evening, it would mean a great deal if you took a moment to like this video and subscribe to the channel. It helps more than you might expect, and it helps this channel continue making exactly this kind of content. And if you feel like leaving a comment, we would genuinely love to know where in the world you are listening from tonight and what time it is there. It is always interesting discovering where listeners are tuning in from around the world. Now, let us begin. The brain you carry with you, the one doing the work of reading or listening right now, weighs approximately 1.3 to 1.4 kilograms, about the weight of a small cantaloupe. It is approximately 73% water. It is soft and grey-pink and unremarkable in appearance, and it contains approximately 86 billion neurons, nerve cells that process and transmit information, along with approximately 85 billion additional supporting cells called glial cells. These neurons are connected to one another by approximately 100 trillion synapses, connection points where one neuron communicates with another through chemical signals. 100 trillion connections. The number of possible combinations of neural connections in a human brain exceeds 10 to the power of 1 million, a number so vast that it exceeds the number of atoms in the observable universe by an almost incomprehensible margin. And despite representing only approximately 2% of your body weight, your brain consumes approximately 20% of your body's total energy. It runs on roughly 12 watts of power, about the same as a very dim light bulb. Yet its information processing capacity is estimated at approximately 1 quadrillion operations per second. Researchers believe that the pattern of neural connections in your brain is unique to you. No two brains, not even those of identical twins, are identical. Your brain is a biological record of your life. Every experience you have ever had, every skill you have ever learned, every person you have ever known, every book you have ever read, all of it has left a physical trace in the structure of your neural connections. You are, in the most literal sense, made of your history. Tonight, we are going to travel through this extraordinary organ slowly and without hurry. We will begin with the history of how humans came to understand the brain, a story spanning thousands of years of curiosity, careful observation, and gradual illumination. We will explore the brain's physical geography, the microscopic language of its neurons, the chemistry of its neurotransmitters, and the remarkable phenomenon of its lifelong capacity to change. We will visit memory, sleep, emotion, perception, and consciousness. We will look at what happens when the brain is at rest and at the deep mysteries that remain unsolved even now. The brain that is listening to these words right now, that is processing them and storing fragments of them and generating the experience of understanding them, is doing all of this while also regulating your breathing, monitoring your body temperature, coordinating your heartbeat, and beginning gently the process of preparing you for sleep. Let us keep it company on that journey. This is not an exaggeration. The ancient Egyptians, one of the most sophisticated civilizations of the ancient world, believed that the heart was the seat of intelligence, emotion, and the soul. When preparing bodies for the afterlife through mummification, Egyptian embalmers carefully preserved the heart as a sacred organ. The brain was removed through the nasal passage using a metal hook and discarded. It was considered of no particular significance. The brain had to wait several thousand years for its importance to be recognized. It was the ancient Greek physician Hippocrates, working in approximately 400 BCE, who made one of the earliest and most consequential proposals in the history of neuroscience. The brain, he argued, was the organ of the mind. Mental illness was not a visitation from the gods, but a disorder of the brain. His contemporary Aristotle disagreed, maintaining that the heart was the seat of the soul and that the brain served merely to cool the blood. Hippocrates turned out to be right, but the debate would continue for centuries. The Roman physician Galen, working in the 2nd century CE, conducted extensive anatomical studies of the brain and nervous system, largely through dissections of animals. He identified the brain as the center of intelligence and the nervous system as the pathway of sensation and movement. His ideas dominated medical thinking for over 1,000 years. In the 17th century, René Descartes proposed that the pineal gland, a small structure deep in the brain, was the seat of the soul, the point at which the immaterial mind met the material body. We now know that the pineal gland produces melatonin and helps regulate sleep-wake cycles. Its role in the soul remains unestablished. The understanding of the brain's electrical nature came in the 1780s, when Luigi Galvani discovered that electrical stimulation could cause muscle contractions in frogs. This was a first glimpse of the electrical language that the nervous system uses to communicate. The implications would take another century to be properly understood. The 19th century brought an era of remarkable localization discoveries. In 1861, a French surgeon named Paul Broca examined the brain of a patient who had spent years unable to produce intelligible speech, despite apparently understanding language. When the patient died, Broca examined his brain and found damage to a specific region of the left frontal lobe. This region, now called Broca's area, was the first brain region to be identified with a specific cognitive function. 13 years later, in 1874, a German neurologist named Karl Wernicke identified a different region in the left temporal lobe, associated with language comprehension. Damage to this region produces a different kind of language disorder, fluent but meaningless speech. These discoveries of Broca and Wernicke established that different brain regions have different functions, a foundational principle of neuroscience that continues to guide research today. The following decades brought the tools needed to see neurons directly. Camillo Golgi developed a silver staining technique in 1873 that made individual neurons visible under a microscope for the first time. Santiago Ramon y Cajal used Golgi's technique with extraordinary patience and artistic skill to establish what is now called the neuron doctrine. The nervous system is composed of discrete individual cells, neurons. They communicate across gaps rather than forming a continuous network. Their shared Nobel Prize in 1906 marked the beginning of modern neuroscience. In 1924, a German psychiatrist named Hans Berger attached electrodes to the human scalp and recorded measurable electrical activity from the brain, the first electroencephalogram, or EEG. He kept the discovery secret for 5 years, fearing ridicule before publishing. The brain, it turned out, produces its own electrical rhythms, detectable from outside the skull. The mid-20th century brought a discovery that would become one of the most studied cases in the history of brain science. Henry Molaison, known for decades only as Patient HM, underwent surgery in 1953 to treat severe epilepsy. The surgeon removed his hippocampus, a seahorse-shaped structure deep in the temporal lobe, on both sides. The epilepsy improved, but something else changed entirely. Molaison could no longer form new, long-term memories. He could hold a conversation normally, but within minutes, he would forget it had occurred. He could recognize his doctors' faces, but not learn their names. Every morning was, for him, a fresh beginning with no memory of the day before. His case revealed something fundamental. The hippocampus is not where memories are stored. His old memories were largely intact, but it is essential for forming new ones. It was a tragic natural experiment that illuminated the architecture of memory more clearly than decades of other research had managed. In 1973, a cellular mechanism for learning and memory was identified: long-term potentiation, or LTP, the persistent strengthening of synaptic connections following repeated activation. And in the 1990s, the discovery of neuroplasticity, the brain's capacity to reorganize itself throughout life, overturned what had been considered settled science, that the adult brain was fixed and unchanging. The first functional MRI studies of the brain were conducted in the early 1990s, providing a window into brain activity in living, awake, behaving human beings. The age of brain imaging had arrived, and with it, an acceleration of discovery that continues to this day. We are still in the early chapters of understanding the brain, but the chapters we have written so far have already transformed what it means to be human. The place to begin that understanding is with the brain's physical structure, its geography, its regions, and what each of them does. That is where we are going next. If you were to hold a human brain in your hands, which neuroscientists and neurosurgeons sometimes do, the first thing you would notice is the surface. It is folded, not randomly, but in a consistent and beautiful pattern of ridges and valleys. The ridges called gyri, the valleys called sulci, that give the brain its characteristic wrinkled appearance. This folding is not incidental. It is a solution to an engineering problem. The outermost layer of the brain, the cerebral cortex, is only approximately 2 to 4 millimeters thick, but it needs to be large. If the cortex were flat rather than folded, It would cover approximately 2,500 square centimeters, roughly the size of a pillowcase. Folding it allows this large surface to fit within the limited space of the skull. It is a remarkably compact solution to the problem of fitting a great deal of computing power into a small container. The cerebral cortex is divided into two hemispheres, left and right, connected by a thick bundle of nerve fibers called the corpus callosum. Each hemisphere is further divided into 4 lobes, each with specialized functions. The frontal lobe occupies the front of the brain, the territory behind your forehead. It is the most distinctly human part of the brain, disproportionately large in humans compared to other primates. It contains the prefrontal cortex, associated with planning, decision-making, personality, and impulse control, and the motor cortex, which controls voluntary movement. When you decide to reach for something, or plan your day, or suppress an impulse, or think about the future consequences of a present action, the frontal lobe is centrally involved. Behind the frontal lobe lies the parietal lobe, which processes sensory information arriving from the body: touch, pressure, temperature, pain, and the sense of where your body parts are in space. The parietal lobe contains the somatosensory cortex, which receives signals from the skin and muscles and creates the brain's map of the body's surface. Below the parietal lobe on each side is the temporal lobe, which is involved in hearing, language comprehension, and memory. It contains the hippocampus and amygdala as medial structures that lie deep within it and that we will visit shortly. The temporal lobe also contains the fusiform face area, a region specialized for recognizing faces, one of the most socially critical visual tasks the human brain performs. At the back of the brain sits the occipital lobe, the brain's primary visual processing center. Despite the fact that seeing feels effortless, the visual system is one of the most computationally demanding things the brain does. Approximately 1/3 of the cerebral cortex is devoted to visual processing. Damage to the occipital lobe can cause blindness even when the eyes themselves are entirely intact, because it is the brain, not the eyes, that actually sees. Deep within the brain, beneath the cerebral cortex, lies the limbic system, a set of structures that govern emotion, memory, and motivation. The hippocampus, named for its resemblance to a seahorse, plays a critical role in forming and consolidating new memories. It is, in a sense, the brain's memory gateway, the structure through which new experiences must pass if they are to become lasting memories. Alongside the hippocampus sits the amygdala, an almond-shaped structure that processes emotions, particularly fear and anxiety. The amygdala can trigger an emotional response before the cortex has fully processed the situation, a rapid automatic system that sometimes responds before conscious awareness catches up. Acting as the brain's relay station is the thalamus, a structure that routes sensory information from the eyes, ears, skin, and other senses to the appropriate cortical areas. Nearly all sensory signals, except those from the nose, pass through the thalamus on their way to conscious processing. Beneath the thalamus is the hypothalamus, tiny, only the size of an almond, yet responsible for regulating hunger, thirst, body temperature, sleep-wake cycles, and hormonal function. It is one of the brain's most powerful structures per unit of volume. At the back and bottom of the brain sits the cerebellum, Latin for little brain. Despite its modest size, relative to the cerebral cortex, the cerebellum contains approximately 69 billion neurons, nearly as many as the rest of the brain combined. Its primary function is coordinating movement, balance, and fine motor control. When you perform a complex physical skill with fluency, catching a ball, typing, playing an instrument, the cerebellum is orchestrating the precise timing and coordination of the required movements. Below the cerebellum, connecting the brain to the spinal cord, is the brainstem. This evolutionarily ancient structure controls the vital automatic functions that keep you alive. Breathing, heart rate, blood pressure, swallowing, and the basic regulation of consciousness. You do not decide to breathe. The brainstem handles it. This evolutionary layering, brainstem oldest, limbic system in the middle, cortex most recent, reflects the brain's history. The structures at the base are shared with most vertebrates. The limbic system is shared with other mammals. The expansive cerebral cortex, and especially the prefrontal cortex, is most fully developed in humans. The brain carries within it traces of its long evolutionary journey. And running through all of these structures is a distinction between two types of tissue. Gray matter, composed of neuron cell bodies and their branching dendrites, is where information is processed. White matter, composed of myelinated axons, is where information travels between regions. The brain is built from processing nodes connected by high-speed communication lines. The communication that happens along those lines, the electrical and chemical language of neurons, is where we are going next. Every thought you have ever had, every memory you carry, every movement you have made, every emotion you have felt, all of it has been produced by the activity of neurons. These remarkable cells are the fundamental units of brain function, and understanding them is the foundation for understanding everything else the brain does. A neuron has three main components. The cell body, or soma, contains the nucleus and the machinery for the cell's survival. Extending from the cell body are dendrites, branching tree-like projections that receive signals from other neurons. And extending in the other direction is the axon, a single long projection that carries signals away from the cell body toward other neurons. A neuron receives incoming signals through its dendrites. These signals arrive from thousands of other neurons. Any given neuron can receive input from between 1,000 and 10,000 other neurons simultaneously. The cell body integrates all of these incoming signals, some of them pushing the neuron toward firing, some of them pushing it away from firing. If the combined input reaches a threshold, the neuron fires. When a neuron fires, it generates what is called an action potential, an electrical signal that travels along the axon to its terminals. The action potential is all or nothing. A neuron either fires completely or does not fire at all. There is no half fire, no weak signal or strong signal, just fire or no fire. The brain encodes information not in the size of individual signals, but in the pattern and frequency of firing across networks of neurons. Signals travel along axons at different speeds depending on whether the axon is myelinated. Myelin is a fatty substance produced by glial cells called oligodendrocytes that wraps around axons in segments, like insulation on an electrical wire. Between segments of myelin are small gaps called nodes of Ranvier, and the electrical signal jumps from node to node rather than traveling continuously along the entire axon. This saltatory conduction, from the Latin for jumping, dramatically increases both the speed and the energy efficiency of signal transmission. Unmyelinated axons conduct signals at approximately 0.5 meters per second. Fully myelinated axons can conduct at up to 120 meters per second, a 240-fold increase in speed. The brain's white matter, the vast network of myelinated axons connecting different brain regions, contains approximately 150,000 kilometers of these high-speed connections. When an action potential reaches the end of the axon, it arrives at the synapse, the connection point between one neuron and another. The synapse is extraordinarily small, approximately 20 to 40 nanometers in width, far too small to be seen with the naked eye. And yet the human brain contains approximately 100 trillion of them. At the synapse, the electrical signal is converted into a chemical signal. The action potential causes small packages of chemical messengers, neurotransmitters, to be released from the sending neuron into the synaptic cleft. These neurotransmitters drift across the gap and bind to receptor molecules on the receiving neuron, either exciting it, making it more likely to fire, or inhibiting it, making it less likely to fire. The receiving neuron then converts this chemical signal back into electrical activity, and the process continues. Neurons do not work alone. They work in circuits, networks of neurons that fire together in coordinated patterns to produce perception, thought, emotion, and behavior. And the strength of connections within these circuits is not fixed. It changes with use. The principle underlying this is sometimes called Hebbian learning, after the psychologist Donald Hebb, who proposed it in 1949. Neurons that fire together wire together. When two neurons repeatedly activate at the same time, the synaptic connection between them is strengthened. When they rarely or never activate together, the connection weakens. This strengthening and weakening of synaptic connections is the cellular basis of learning and memory. The mechanism of synaptic strengthening, long-term potentiation, or LTP, involves the insertion of more receptor molecules into the postsynaptic membrane, making the connection more sensitive and more reliable. Over time, repeated activation can lead to the physical growth of new synaptic contacts, further strengthening the circuit. Supporting all of this neural activity is an extensive supporting cast of non-neuronal cells called glial cells. Astrocytes regulate the chemical environment surrounding synapses, take up excess neurotransmitters, and modulate synaptic transmission. They are active participants in brain communication, not merely passive scaffolding. Oligodendrocytes produce the myelin that speeds signal transmission. Microglia, the brain's resident immune cells, patrol the brain for damage and infection. And microglia do something else particularly important during development. They prune unused synaptic connections. Synaptic pruning is the process by which the brain eliminates connections that are not being actively used. It is particularly intense during adolescence, the brain reducing its total number of connections as part of a refinement process. The connections that survive are those that have been used and strengthened by experience. Those that have not been used are removed, a neural expression of use it or lose it. The result of all this, the firing of action potentials, the release of neurotransmitters, the strengthening and pruning of synaptic connections, is a brain that continuously rewires itself in response to what it experiences. But before we explore how that rewiring happens over time, the phenomenon of neuroplasticity, it is worth understanding more about the chemical dimension of neural communication. The brain does not speak only in electrical signals. It also speaks in chemistry. That chemistry is where we are going next. Every moment of your experience, every mood, every motivation, every feeling of calm or restlessness, every flicker of pleasure or anxiety is shaped in part by chemistry, not in a reductive way that diminishes the richness of experience, but in a real and fascinating way. The brain communicates with itself through chemical messengers, and those messengers are among the most consequential molecules in the known universe. These chemical messengers are called neurotransmitters. They are produced by neurons and released into the synaptic cleft, the microscopic gap between neurons, where they bind to receptors on the receiving cell and influence whether it fires. Different neurotransmitters have different effects. Some excite the receiving neuron, making it more likely to fire. Some inhibit it, making it less likely. And some modulate the neuron's response to other signals, adjusting the sensitivity of the receiving cell without directly causing it to fire or not fire. The brain's primary excitatory neurotransmitter is glutamate. It is present at the majority of excitatory synapses throughout the brain and is involved in virtually all brain functions: learning, memory, development, sensation, and cognition. When glutamate binds to its receptors, it increases the likelihood that the receiving neuron will fire. Glutamate is the brain's accelerator, the neurotransmitter that drives neural activity forward. Balancing glutamate is GABA, gamma-aminobutyric acid, the brain's primary inhibitory neurotransmitter. Where glutamate accelerates, GABA brakes. It reduces the likelihood of a neuron firing, damping down activity and promoting stability and calm. Many anti-anxiety medications and sedatives, including benzodiazepines and alcohol, work by enhancing GABA activity. The feeling of relaxation they produce reflects the brain's inhibitory system, gaining the upper hand over its excitatory one. The balance between glutamate and GABA is one of the most fundamental features of brain function. Too much glutamate activity without sufficient GABA can lead to seizures, uncontrolled neural firing. Too much GABA activity can produce excessive sedation. The brain walks a continuous tightrope between these two forces, maintaining a level of activity appropriate to the demands of the moment. Dopamine is perhaps the most widely discussed neurotransmitter and one of the most frequently misunderstood. It is often described as the pleasure chemical, but this is an oversimplification. Dopamine is more precisely a motivation and anticipation chemical. It is released not only when you experience a reward, but, and this is the crucial point, when you anticipate one. The brain's reward system releases dopamine in response to cues that predict rewarding outcomes, driving you toward those outcomes before they arrive. The nucleus accumbens, a key structure in the brain's reward circuit deep in the basal forebrain, is central to this dopamine-driven motivational system. When you feel the pull towards something pleasurable, food, social connection, a satisfying task, a piece of music you love, dopamine is part of the mechanism creating that pull. Interestingly, curiosity itself activates the dopamine system. Learning new information produces a dopamine response similar to other rewards. The brain treats the acquisition of knowledge as intrinsically valuable, which may explain why genuine curiosity feels energizing rather than effortful, and why tonight's journey through neuroscience might feel, in small ways, genuinely satisfying rather than merely informative. Serotonin influences mood, sleep, appetite, and social behavior. Its relationship with mental well-being is complex. Most antidepressant medications work by increasing the availability of serotonin in synapses, though the relationship between serotonin levels and mood is not a simple one-to-one correspondence. Serotonin appears to play a role in maintaining emotional stability and regulating the sensitivity of emotional responses. Norepinephrine, also called noradrenaline, is the brain's alertness chemical. It promotes wakefulness, attention, and focus, and plays a central role in the stress response. When you encounter something that demands your attention or poses a threat, norepinephrine levels rise, sharpening focus and preparing the brain and body for action. Acetylcholine is involved in muscle activation, learning, memory, and the regulation of the sleep-wake cycle. It is the neurotransmitter that carries signals from motor neurons to muscles, producing movement. In the brain, acetylcholine plays important roles in attention and memory formation, and its systems are significantly affected in Alzheimer's disease. The brain also produces endorphins, naturally occurring pain-relieving chemicals that bind to the same receptors as opioid drugs. Endorphins are released during exercise, sustained physical effort, and emotional stress. The sense of well-being that follows vigorous exercise, sometimes called runner's high, is partly mediated by endorphin release. Oxytocin, produced in the hypothalamus and released by the pituitary gland, is sometimes called the bonding hormone. It promotes trust, social bonding, and prosocial behavior. It is released during positive physical contact, genuine social connection, and moments of intimacy. It plays a particularly important role in the bond between a parent and a newborn child. Researchers believe oxytocin contributes to the sense of warmth and safety that comes from being in the company of people we trust. Cortisol, the brain's primary stress hormone, has a more complex role. In the short term, it is essential. It mobilizes energy, sharpens attention, and prepares the brain for demanding situations. But elevated cortisol over extended periods has measurable negative effects on brain structure, particularly on the hippocampus and prefrontal cortex. It also amplifies the consolidation of emotional memories, making stressful experiences more memorable than neutral ones. Together, these neurotransmitters constitute the chemical vocabulary of the brain, a language in which mood, motivation, alertness, calm, bonding, and memory are all expressed in molecular terms. The words in this vocabulary are being spoken continuously across trillions of synapses, shaping every moment of experience. And that vocabulary is not fixed. It changes with the brain itself in response to experience, learning, and time. That capacity for change is one of the most important discoveries in modern neuroscience. That is where we are going next. This belief turned out to be wrong. The discovery of neuroplasticity, the brain's capacity to reorganize itself by forming new neural connections throughout life, is one of the most consequential findings in the history of brain science. It changed not only what scientists understood about the brain, but what seemed possible in terms of recovery from injury, learning across the lifespan, and the brain's capacity to respond to the demands of experience. Neuroplasticity operates at many levels. At the finest scale, it involves the strengthening or weakening of individual synaptic connections, the cellular mechanism of long-term potentiation described in the previous section. When neurons repeatedly fire together, the connection between them is strengthened. The synapse becomes more sensitive and more reliable. The signal passes more efficiently with each use. This is the microscopic foundation of learning. Every skill practiced, every fact studied, every experience repeated strengthens the neural circuits responsible for that skill, fact, or experience. At a slightly larger scale, neuroplasticity involves the growth of new synaptic contacts between neurons, the physical extension of dendritic branches to form new connections. Learning a new skill does not merely use existing circuits more efficiently. Over time, it creates new structural connections that were not previously there. The brain that has learned something is literally not the same brain that existed before the learning occurred. At the largest scale, neuroplasticity involves the reorganization of entire cortical maps, the regions of the cortex devoted to specific functions shifting in response to sustained experience. Michael Merzenich, whose work in the 1980s and 1990s was central to establishing the reality of adult neuroplasticity, demonstrated that the sensory cortex reorganizes dramatically in response to experience. When a finger is repeatedly stimulated, the cortical area devoted to that finger expands. When a finger is amputated, neighboring fingers' cortical representations expand into the vacated area. The map is not fixed. It is continuously redrawn based on use. The implications of this finding extend well beyond laboratory experiments. Studies of London taxi drivers, who spend years memorizing thousands of streets and their connections, have found greater grey matter volume in the hippocampus than in non-taxi drivers. The hippocampus, involved in spatial navigation and memory, physically grows in response to sustained navigational demands. Studies of musicians have found expanded cortical representation of the fingers used to play their instruments. Studies of bilingual people have found denser grey matter in the parietal cortex compared to monolingual individuals. What we repeatedly do shapes what we neurologically become. Neuroplasticity also operates in the direction of recovery. When a stroke damages a portion of the motor cortex, the result is typically paralysis or weakness on one side of the body. But through intensive rehabilitation, repeated practice of the affected movements, neighboring brain regions can gradually take over some of the functions of the damaged area. The recovery is real, and it is neural. Other parts of the brain literally reorganize to compensate. Plasticity makes rehabilitation possible. Exercise has been found to promote neurogenesis, the growth of new neurons, particularly in the hippocampus. This was once considered impossible. It was believed that adult humans could not grow new neurons. The work of Peter Eriksson, Fred Gage, and their colleagues in 1998, demonstrating adult neurogenesis in the human hippocampus, overturned this belief. New neurons are born in the dentate gyrus of the hippocampus throughout life, and their production is promoted by aerobic exercise, environmental enrichment, and learning, and suppressed by chronic stress and social isolation. Meditation practice has been shown to produce measurable structural changes in the brain. Studies by Sarah Lazar and colleagues at Harvard found that long-term meditators have greater cortical thickness in the prefrontal cortex and anterior insula, regions associated with attention, self-awareness, and interoception. Even relatively brief periods of meditation practice have been found to produce detectable changes in brain structure. Chronic stress, by contrast, works in the other direction. Sustained elevated levels of cortisol, the body's primary stress hormone, damage dendrites in the hippocampus and prefrontal cortex, reducing the volume of these structures over time. Simultaneously, chronic stress promotes dendritic growth in the amygdala, the brain's threat detection center. The structural effect of chronic stress is to shift the brain toward a more reactive, more threat-sensitive, less rationally regulated state. Precisely the changes that make chronically stressed people more emotionally reactive and less able to think clearly. One reason this matters is that it is reversible. Neuroplasticity works both ways. Recovery from chronic stress through rest, exercise, social support, and the reduction of stressors can restore hippocampal and prefrontal volume over time. The brain that has been shaped by stress can be reshaped by recovery. The deepest implication of neuroplasticity is this: the brain is not a passive recipient of experience. It is an active, dynamic, continuously remodeling structure that participates in its own formation. Every choice about how to spend time, what to practice, what to attend to, and how to respond to difficulty is a choice about what kind of brain to have. The brain that has been shaped by all of your experiences thus far, including the experience of listening to this, is the starting point for whatever comes next. And among the most important things that comes next every night is memory consolidation. That is where we are going next. The brain's memory system is not a single structure or a single process. It is a collection of systems, different types of memory involving different brain regions, operating on different timescales, serving different purposes. At the center of the story of memory is the hippocampus, the seahorse-shaped structure deep in the temporal lobe, whose importance was so dramatically revealed by the case of patient HM. The hippocampus is essential for the formation and consolidation of new episodic memories, memories of specific events and experiences, and new semantic memories, memories of facts and general knowledge. It acts as a kind of memory gateway. New experiences must pass through hippocampal processing to become lasting long-term memories. What the hippocampus does is bind together the different elements of an experience that are stored in different parts of the cortex. The visual details of a scene are processed in the visual cortex. The sounds are processed in the auditory cortex. The emotional significance is processed by the amygdala. The hippocampus weaves these distributed pieces together into a coherent memory, the binding function that allows a complete episode to be recalled as an integrated whole. Different types of memory draw on different systems. Procedural memory, the memory for skills and habits, does not depend on the hippocampus. Patient HM, who could not form new episodic or semantic memories, could still learn new motor skills through practice. He would improve at mirror drawing, tracing a shape while watching only its mirror image, from session to session, despite having no memory of having done the exercise before. The cerebellum and basal ganglia are more central to procedural memory. The skill is stored in the doing, not in the remembering of having done. One of the most important facts about memory is that it is not a recording. Memory is a reconstruction. When you remember something, you are not retrieving a stored copy of the original experience, like playing back a video file. You are reconstructing the experience from fragments, some stored in memory, some filled in from general knowledge, some influenced by what you have learned since the original event. The reconstruction is usually accurate in its broad outlines, but it can be subtly altered by subsequent experience, by the emotional state in which you recall it, and by the mere act of recalling it. This has an important implication. Every act of remembering briefly renders a memory vulnerable to modification. When a memory is recalled, it must be restabilized before it is stored again, a process called reconsolidation. During this window, the memory can be updated, altered, or in some circumstances, weakened. Memory is a living process, not a static archive. The distributed nature of memory, stored across networks rather than at single locations, means that memories are resilient to localized damage, but can be degraded by more extensive damage. Losing one piece of the network does not erase a memory. It merely makes it hazier or harder to access, like a hologram that becomes blurrier as pieces are removed. Sleep plays a crucial role in memory consolidation. During slow-wave sleep, the hippocampus replays patterns of neural activity corresponding to experiences from the waking day, essentially reviewing what happened and strengthening the synaptic traces. This replay is thought to be part of the process by which memories are transferred from temporary hippocampal storage to more permanent cortical storage. The hippocampus is not the final address of a memory. It is a temporary processing center, and over time memories are consolidated into distributed cortical networks where they can persist without hippocampal support. Emotional memories are particularly resistant to forgetting. The amygdala, which processes emotional significance, modulates the strength of hippocampal memory consolidation. Emotionally significant events trigger amygdala activation, which releases norepinephrine that enhances synaptic strengthening in the hippocampus. This is why emotionally charged experiences, whether joyful or painful, tend to be remembered more vividly and durably than neutral ones. Cortisol, the stress hormone, also influences memory encoding, making stressful experiences more memorable by similar mechanisms. This is adaptive in an evolutionary sense. Events that were emotionally significant or threatening deserve to be remembered well. The brain prioritizes its memory resources according to emotional salience. Forgetting in contrast to what might seem intuitive, is not simply a failure of memory. It is an active and useful process. The brain actively suppresses and eliminates memories that are no longer relevant, freeing capacity and reducing interference. A brain that remembered everything with equal clarity would be overwhelmed by the weight of its own history. Forgetting is the editing that makes remembering useful. The brain's capacity to store information is estimated at approximately 2.5 petabytes, roughly equivalent to 3 million hours of television. This is not a fixed limit that fills up over a lifetime. Memory storage in the brain does not work like hard drive storage. The brain is not running out of room. What limits our memory is not capacity, but encoding, consolidation, and retrieval. The processes by which experiences become memories and memories become accessible. And one of the most important things that supports all of these processes, encoding, consolidation, and retrieval, is something the brain does every night, whether we ask it to or not. That is where we are going next. There is a common misconception about sleep, that it is a passive state, a withdrawal from the active business of the brain, a kind of biological standby mode. The truth is very nearly the opposite. Sleep is a period of intense and purposeful biological activity in the brain. The sleeping brain is not idle. It is performing work that cannot be done during wakefulness. Maintenance, consolidation, clearance, and preparation. The importance of sleep to brain function is not a minor footnote, but a central fact about how the brain sustains its ability to operate. To understand what happens in the brain during sleep, it helps to understand that sleep is not uniform. It proceeds through distinct stages, each characterized by a different pattern of electrical activity that is visible on an electroencephalogram. The transition from wakefulness to sleep begins with the emergence of alpha waves, rhythmic oscillations at approximately 8 to 12 hertz that appear when the eyes are closed and the mind is relaxed but still awake. As sleep deepens, alpha waves give way to theta waves, slower oscillations at approximately 4 to 8 hertz, characteristic of light sleep and the transitional state between waking and sleeping. Deeper still is slow wave sleep, also called delta sleep or non-REM stage 3, characterized by large, slow delta waves at less than 4 hertz. This is the deepest and most restorative stage of sleep. The body is deeply relaxed, the brain's metabolic rate is reduced, and yet something important is happening. During slow wave sleep, the hippocampus replays patterns of neural activity corresponding to experiences from the waking day. This replay is not random. It appears to reflect the structured retransmission of daytime experiences from the hippocampus to the neocortex, the process by which memories are transferred from temporary hippocampal storage to more permanent cortical storage. Sleep spindles, bursts of oscillatory activity at approximately 12 to 15 hertz appearing during non-REM sleep, have been associated with this memory consolidation process and with individual differences in memory performance and intelligence. After a period of slow wave sleep, the brain enters a dramatically different state, rapid eye movement sleep, or REM sleep. During REM, the eyes move rapidly beneath closed lids. Dreaming occurs most vividly during this stage, and the brain's electrical activity during REM sleep closely resembles its activity during alert wakefulness. The sleeping brain is, in some respects, as active as the waking brain. REM sleep appears particularly important for emotional memory processing and for the integration of new information with existing knowledge. During REM, the brain seems to find connections between recently acquired information and previously stored knowledge, one mechanism by which sleep contributes to insight and creativity. People who sleep after learning perform significantly better than those who remain awake for an equivalent period. In 2013, a discovery transformed understanding of why sleep is necessary. Mike and Nedergaard and colleagues at the University of Rochester described the glymphatic system, a network of channels surrounding blood vessels in the brain that serves as the brain's waste clearance mechanism. During sleep, the brain's cells shrink by approximately 60%, allowing cerebrospinal fluid to flow more freely through the glymphatic channels and flush out metabolic waste products that accumulate during waking neural activity. Among the waste products cleared by the glymphatic system is amyloid beta, a protein that, when it accumulates excessively, is associated with Alzheimer's disease. The glymphatic system is approximately 10 times more active during sleep than during wakefulness. A brain that consistently does not sleep enough is a brain whose waste products accumulate, with potentially significant long-term consequences for health. Sleep deprivation has measurable effects on brain function that are visible on brain imaging. Reduced sleep impairs activity in the prefrontal cortex, the region most associated with rational decision-making, impulse control, and planning, while increasing the reactivity of the amygdala. A sleep-deprived brain is one that is less capable of calm reasoning and more prone to emotional reactivity. The effects of a single night of poor sleep on cognitive performance are comparable to mild alcohol intoxication. The brain's sleep-wake cycle is coordinated by the suprachiasmatic nucleus, a cluster of approximately 20,000 neurons in the hypothalamus that acts as the brain's master clock. It receives light information from the retina and uses it to synchronize the body's circadian rhythms, the approximately 24-hour cycles of physiology and behavior. As darkness falls and melatonin, produced by the pineal gland in response to the suprachiasmatic nucleus's signals, accumulates in the bloodstream, the drive to sleep increases. The mystery of why we dream, what purpose dreams serve, has not been definitively resolved despite extensive research. Various proposals have been made. Dreams may be involved in emotional processing, allowing the brain to work through difficult experiences in a relatively safe context. They may be a byproduct of memory consolidation, as the hippocampus replays the day's experiences and the narrative-building machinery of the cortex generates stories from the fragments. They may involve threat simulation, rehearsing responses to challenging scenarios. No single theory has proven sufficient, and the question of why the brain generates the vivid narrative experiences of dreaming remains genuinely open. What is not in question is the profound importance of sleep to brain function. The glymphatic system begins its work. Memory consolidation proceeds. The brain's electrical rhythms slow and deepen, and the prefrontal cortex, exhausted from the day's demands, has the opportunity to restore its capacity for tomorrow. What is happening in the brain's emotional systems, both during sleep and during the waking day, is equally fascinating. That is where we are going next. There is an old and persistent idea that emotion and reason are opposites, that the emotional brain and the rational brain are in competition, and that the best thinking happens when emotion is set aside. This idea is wrong, and the neuroscience of emotion has spent several decades demonstrating why. Emotion is not the enemy of cognition. It is woven through all cognition. Feeling and thinking are not separate processes happening in different brain regions that occasionally interfere with each other. They are aspects of the same neural processing, mutually dependent and continuously interacting. A brain without emotion would not be a more rational brain. It would be a brain incapable of making decisions at all. The evidence for this comes from patients with damage to the orbitofrontal cortex, a region of the prefrontal cortex involved in integrating emotional signals with decision-making. These patients score normally on standard tests of intelligence, reasoning, and memory. They can discuss the pros and cons of different options logically and coherently, but they cannot make good decisions. Faced with real choices, where to live, how to manage relationships, what to do with their time, they flounder, unable to settle on one option over another, or making choices that are clearly counterproductive. The neuroscientist Antonio Damasio, who studied many such patients, proposed the somatic marker hypothesis that emotions serve as rapid evaluative signals that guide decision-making. When you consider an option, emotional memory systems generate a fast, intuitive sense of whether it is good or bad, a body-based signal, a feeling. This feeling is not infallible, but it is essential. Without it, the purely rational evaluation of options becomes paralyzed by its own completeness. At the center of the brain's emotional processing is the amygdala, the almond-shaped structure in the medial temporal lobe that processes fear, anxiety, and threat. The amygdala receives sensory information through a rapid, direct pathway from the thalamus before that information has been fully processed by the cortex. This allows the amygdala to generate an emotional response, a jolt of fear, a tensing of the body, before conscious awareness has caught up with what is happening. This rapid automatic response was adaptive in the evolutionary environment in which it developed. If a predator appeared, waiting for full cortical analysis of the situation before responding was dangerous. The amygdala's fast approximate evaluation, is this safe or not, could save a life, even if its assessment was sometimes inaccurate. In the modern environment, the same rapid amygdala response still operates, generating fear and anxiety responses to perceived threats, including social threats, before conscious evaluation occurs. The amygdala does not distinguish very precisely between a physical threat and a social threat. Both trigger similar responses. Which brings us to one of the more poignant findings in social neuroscience. Social pain, the experience of rejection, exclusion, or social loss, activates some of the same brain regions as physical pain. The dorsal anterior cingulate cortex and anterior insula, both involved in the processing of physical pain, show increased activity when a person is socially excluded. The brain treats being left out as a kind of injury. The experience of heartbreak, of social rejection, of loneliness, these are not merely metaphorically painful. Neurologically, they share circuitry with physical hurt. The brain has a negativity bias, a tendency to process negative stimuli more deeply, respond to them more strongly, and remember them more durably than positive stimuli of comparable intensity. This bias is thought to reflect evolutionary prioritization. In an environment where threats were real and consequential, attending more strongly to negative information was protective. A missed opportunity was costly. A missed threat could be fatal. The brain is still calibrated for that ancestral environment, processing bad news more thoroughly than good. Chronic stress has measurable and lasting effects on the emotional brain. Sustained elevated cortisol, the primary stress hormone, damages dendritic connections in the hippocampus and prefrontal cortex, physically shrinking these structures over time. Simultaneously, it promotes dendritic growth in the amygdala. The structural effect is a brain shifted toward greater emotional reactivity and reduced capacity for rational regulation. A self-reinforcing cycle in which stress makes the brain more reactive to stress. But the brain's emotional systems are also shaped by positive experience and deliberate practice. The prefrontal cortex exerts top-down regulatory control over the amygdala. When the prefrontal cortex is active, it can modulate the amygdala's response, reducing the intensity of emotional reactions and creating the space for a considered response rather than an automatic one. This capacity for emotional regulation, the ability to feel an emotion without being entirely governed by it, is mediated by the strength of prefrontal amygdala connections. And like other neural circuits, these connections are strengthened by use. Practices that repeatedly engage the prefrontal cortex's regulatory capacity, including mindfulness meditation, have been shown to increase the density of prefrontal amygdala connections, reducing amygdala reactivity over time. Emotional regulation is not simply a matter of willpower. It is a skill that changes the brain when practiced. The role of emotion in memory adds another dimension. Emotionally significant experiences are remembered more vividly and durably than neutral ones, because the amygdala enhances hippocampal memory consolidation for events of emotional importance. What we care about, we remember, and what we remember, in turn, shapes how we feel about similar experiences in the future. Emotion and memory are in continuous dialogue, each shaping the other. Understanding emotion as a brain process, as something produced by neural circuits that can be influenced, trained, and shaped by experience, changes the relationship between a person and their emotional life. Feelings are not simply things that happen to us. They are things our brains produce, and the brain is changeable. What the brain produces in the way of experience goes beyond emotion. It constructs the entire reality in which we seem to live. That construction is where we are going next. There is a temptation to think of perception as straightforward, as the simple reception of information from the world. Light enters the eye and is registered. Sound enters the ear and is heard. The world impresses itself on the senses, and the brain records what is there. This picture is wrong in ways that are both surprising and deeply consequential. Perception is not a passive reception of reality. It is an active construction. The brain does not simply register what is in the world. It builds a model of the world from incomplete, noisy, and often ambiguous sensory information, filling in gaps, making inferences, and generating a rich, coherent experience from inputs that would, on their own, support many different interpretations. The most influential framework for understanding this is called predictive processing, sometimes called the predictive coding framework. On this account, the brain is fundamentally a prediction machine. At every moment, it generates predictions about what it expects to perceive, based on past experience, current context, and the body's state. And it compares these predictions to the incoming sensory signals. The incoming signals serve not to build perception from scratch, but to update and refine the predictions. When the incoming signal matches the prediction, little updating is needed. The prediction is confirmed and perception proceeds smoothly. When the signal diverges from the prediction, when something unexpected happens, the brain updates its model, revising its predictions for the future. Perception is the result of the brain's best current guess about what is causing its sensory inputs. This framework explains many otherwise puzzling phenomena. Optical illusions work by presenting information that systematically misleads the brain's predictions. The brain applies its usual assumptions and generates an incorrect percept. The same physical stimulus can be perceived differently depending on context, expectation, and prior experience, because perception is always a mixture of bottom-up sensory input and top-down prediction. The visual system alone illustrates the complexity of perception. Information travels from the retina along the optic nerve to the primary visual cortex in the occipital lobe, and from there, it is distributed through at least two major parallel processing streams. The ventral stream, running from the occipital lobe towards the temporal lobe, processes object identity, what something is. The dorsal stream, running from the occipital lobe toward the parietal lobe, processes spatial location and guides action, where something is and how to interact with it. These streams operate largely in parallel, and damage to one can impair object recognition while leaving spatial processing intact, or vice versa. Despite this parallel processing, the result of all these distributed computations is a unified, seamless, perceptual experience. You do not experience separate streams of visual processing. You experience one coherent visual world. How the brain integrates information processed in different regions and at different times into a single unified percept is called the binding problem, and it remains one of the most challenging unsolved questions in neuroscience. Pain provides one of the clearest illustrations of the brain's constructive role in perception. Pain is not simply a signal that travels from damaged tissue to the brain, where it is passively registered. Pain is constructed by the brain, an interpretation of bodily signals that is influenced by context, expectation, attention, and meaning. The same injury can produce dramatically different amounts of pain depending on the circumstances. A soldier wounded in battle may feel little pain in the immediate aftermath. A person anticipating a painful procedure may experience more pain than the same procedure would cause without anticipation. People who believe a painkiller will work typically experience greater pain relief than those who do not, even when the substance is inert. These are not simply psychological effects overlaid on a physical signal. They reflect the brain's active construction of the pain experience from multiple inputs. Phantom limb pain, in which amputees experience pain in a limb that no longer exists, makes this construction particularly vivid. There is no tissue to be damaged, no peripheral signal to transmit. The pain is generated entirely by the brain's continued representation of the missing limb. The body map in the brain persisting and sometimes generating pain signals in a region that the peripheral nervous system no longer serves. All of this, the construction of perception, the integration of sensory streams, the generation of pain and pleasure and color and sound, is accompanied by something that neuroscience has found it extraordinarily difficult to explain: consciousness, the subjective experience of perceiving, thinking, and feeling, the sense that there is something it is like to be you, having these experiences right now. The hard problem of consciousness, a term coined by the philosopher David Chalmers, is the question of why and how subjective experience arises from neural activity. The easy problems, explaining attention, perception, memory, behavior, are in principle solvable by neuroscience. We can, in principle, explain how the brain processes information and produces outputs. What we cannot yet explain is why any of this processing is accompanied by experience. Why there is something it is like to be a brain doing these things, rather than merely information processing happening in the dark. No current theory of neuroscience adequately answers this question. The neural correlates of consciousness, the brain regions and processes most closely associated with conscious awareness, are actively studied and debated. The claustrum, a thin sheet of neurons beneath the insular cortex, was proposed by Francis Crick and Christoph Koch as a possible integrating center for consciousness. Other researchers have emphasized the role of thalamocortical loops, or of specific patterns of neural communication across the cortex. The mystery remains, and it is perhaps the most intimate mystery in science, because whatever consciousness is, you are having it right now. When that consciousness is not focused on anything in particular, when the mind is at rest, the brain does not go quiet. That resting brain is where we are going next. In 2001, a neuroscientist named Marcus Reichel and his colleagues at Washington University published a paper that changed how scientists think about the resting brain. They had noticed something unexpected in their brain imaging data. When research participants were given demanding cognitive tasks requiring focused attention and deliberate mental effort, certain brain regions consistently deactivated. And when the participants were simply resting, doing nothing in particular, those same regions consistently activated. The regions that lit up during rest formed a consistent, reproducible network. Reichel called it the default mode network. The discovery was striking for several reasons. First, it suggested that the brain has a characteristic mode of operation during rest, a default pattern of activity that emerges whenever the brain is not engaged in externally directed tasks. Second, it implied that rest is not the absence of brain activity, but a different kind of brain activity, with its own structure, its own functions, and its own importance. The default mode network includes the medial prefrontal cortex, the posterior cingulate cortex, the precuneus, and the angular gyrus, regions distributed across the brain that consistently activate together during wakeful rest. what are these regions doing? Researchers believe the default mode network is associated with self-referential processing, thinking about oneself, one's past experiences, and one's future plans. It is active during mind wandering, the spontaneous, undirected flow of thought that occurs when attention is not anchored to an external task. It appears to be involved in imagining other people's mental states. Theory of mind. It plays a role in creative thinking and in the generation of novel associations between ideas. It is, in a sense, the neural substrate of the inner life. The brain at rest is not doing nothing. It is doing something different, something that appears to be essential to the full functioning of cognition. Mind wandering, which is easy to dismiss as unproductive distraction, may serve important functions, consolidating memories, processing social information, simulating future scenarios, and generating the creative connections that sometimes produce insight. One of the most intriguing features of the default mode network is how much energy the resting brain consumes. The brain at rest uses approximately 80% of its total energy budget. Only an additional 0.5 to 1% of energy is required for even demanding cognitive tasks. The resting brain is already running at nearly full capacity. What that resting activity is doing with all that energy is still not entirely understood. Flow states, periods of total absorption in a challenging activity, offer a different kind of insight into the resting brain. Flow, as described by Mihaly Csikszentmihalyi, is characterized by effortless performance, loss of self-consciousness, and a sense of complete engagement. It tends to occur when a person's skill level is well matched to the challenge of the task. Not too easy, not too overwhelming. Brain imaging studies of flow states suggest they are associated with reduced activity in the prefrontal cortex, the region most associated with self-monitoring, planning, and conscious control. This is called the transient hypofrontality hypothesis. During flow, the brain appears to reduce the activity of its most deliberate and self-aware systems, allowing more automatic and efficient processing to operate without constant conscious oversight. The result is performance that feels effortless precisely because it has been partially relieved of the weight of self-consciousness. As wakefulness gives way to rest, the brain's electrical rhythms shift. Alpha waves, rhythmic oscillations at approximately 8 to 12 Hz, are the signature of relaxed wakefulness. They appear when the eyes are closed and the mind is calm but not yet asleep. They reflect a brain that is awake but not actively engaged with external demands, a state of alert restfulness that is distinct from both focused attention and deep sleep. As sleep approaches, alpha waves give way to theta waves, slower oscillations at approximately 4 to 8 Hz, the characteristic rhythm of the transition between waking and sleep. This transitional state has been associated with a particularly creative form of cognition, one in which the normal constraints of logical sequential thinking relax and unusual associations become more available. Many people report hypnagogic experiences during this state, fragmentary images and ideas that arise as the mind drifts between waking and sleep. The relaxation response, first described by Herbert Benson, is a physiological state that can be intentionally elicited through quiet, restful attention. Practices including meditation, deep breathing, Progressive muscle relaxation and gentle movement produce a consistent pattern of physiological changes. Decreased heart rate, reduced blood pressure, lower cortisol levels, and increased alpha wave amplitude. The body and brain shift toward a state of calm restoration. This state is not passive. It is an active biological mode, one that supports the repair and restoration of systems taxed by the demands of wakefulness. The brain, given the opportunity to enter this state, takes advantage of it. The brain settling into rest right now, as this section draws to a close, is making that transition. The default mode network is taking precedence over the task-focused networks. Alpha waves are beginning to emerge. The systems of focused attention are beginning to hand over to the systems of quiet inner processing. The resting brain has much to say about the brain's other remarkable capacities for language, music, and the extraordinary complexity of social life. That is where we are going next. Language is one of the most distinctly human capacities, a system so natural to us that we rarely pause to appreciate what an extraordinary achievement it represents. A child of 2 is already producing novel sentences they have never heard before, following grammatical rules they have never been explicitly taught. By the age of 6, the average child has a vocabulary of approximately 14,000 words and a command of the complex grammatical structures of their native language that took linguists decades to formally describe. The brain systems underlying language were among the first cortical functions to be localized in the classic work of Broca and Wernicke in the 19th century. Broca's area in the left inferior frontal gyrus is involved in speech production and grammatical processing. Damage to it produces Broca's aphasia. The patient understands language and knows what they want to say, but cannot produce fluent speech. Sentences come out telegraphically reduced, stripped of their grammatical structure. Wernicke's area in the left posterior superior temporal gyrus is involved in language comprehension. Damage produces Wernicke's aphasia, fluent speech that has lost its meaning. The sentences are grammatically formed, but semantically empty. Words strung together in plausible order, but conveying nothing. The patient does not know that their speech is nonsensical. These classical aphasia syndromes demonstrate that production and comprehension of language are dissociable. They depend on different brain regions, but the full language network is considerably more extensive than these two areas. It includes the angular gyrus, which integrates visual and auditory information with language, supporting reading. It includes the anterior temporal lobe, which processes meaning. It includes the supplementary motor area and premotor cortex, which plan the motor sequences of speech. Language is a distributed system, not a localized capacity, but a coordinated network. Language also has a biological predisposition. Noam Chomsky proposed in the 1950s and 1960s that humans have an innate language acquisition device, a biological foundation that makes the learning of grammatical structure rapid and effortless in children in a way that would not be possible if language learning were entirely environmental. While the details of this proposal have been extensively debated and refined, the basic insight remains influential. The human brain is biologically prepared for language in ways that other brains are not. Music engages the brain in a particularly broad and deep way. When you listen to music, you find meaningful. The auditory cortex processes the sound. The motor cortex and cerebellum respond to the rhythm. The limbic system generates emotional response. The hippocampus retrieves musical memories, and the nucleus accumbens releases dopamine in response to pleasurable passages. Music is one of the most broadly activating experiences available to the brain, simultaneously engaging sensory, motor, emotional, and reward systems that rarely operate together in this coordinated way. The motor system's activation during music listening, even when the listener is not moving, is particularly interesting. Researchers believe it underlies the almost universal tendency for music to induce movement, tapping, swaying, dancing. The motor system is recruited by rhythmic sound even without any intention to move, perhaps because rhythm in the auditory world has historically been associated with movement in the physical world. Music can trigger the release of dopamine, the neurotransmitter associated with reward and anticipation, in response to particularly pleasurable musical moments. The chills or goosebumps that many people experience when hearing music they find especially moving are associated with dopamine release, the brain responding to music as a genuine reward, not merely a pleasant sensation. Mirror neurons were discovered in macaque monkeys in the early 1990s by Giacomo Rizzolatti and colleagues at the University of Parma, who noticed that certain neurons fired both when the monkey performed an action and when it watched another individual perform the same action. In humans, mirror neuron-like activity, observable through neuroimaging, is implicated in the understanding of others' intentions, in imitation, and in empathy. When you watch someone else in pain, some of the same neural circuits activate as when you experience pain yourself. The brain partially simulates others' experiences, which is the neural mechanism that makes empathy possible. The fusiform face area, a region in the temporal lobe, is specialized for recognizing faces. The human brain devotes remarkable processing resources to face recognition. We are extraordinarily good at it, capable of recognizing familiar faces across years of aging, dramatic lighting changes, and partial occlusion. We also tend to see faces in ambiguous stimuli, clouds, wood grain, toast, demonstrating that face detection systems are calibrated to be highly sensitive even at the cost of occasional false positives. The sensory homunculus, first mapped by Wilder Penfield, is the brain's map of the body's surface in the somatosensory cortex. It is a distorted map. The hands and face are represented by disproportionately large cortical areas, while the trunk and legs are represented by relatively small ones. The distortion reflects the density of sensory receptors. The hands and face are the most sensitive parts of the body, requiring the most cortical space to process their signals with adequate resolution. Neural coupling is a measurable phenomenon in which the brain activity of a speaker and a listener synchronize during communication. Uri Hassan and colleagues at Princeton demonstrated that the listener's brain activity comes to resemble the speaker's, with better coupling associated with better comprehension and richer communication. When two people are truly communicating, when meaning is being conveyed, their brains, in a measurable sense, are becoming more similar. The social brain, The extensive cortical machinery devoted to navigating relationships, understanding others, and communicating meaning is one of the most distinctive features of the human nervous system. A substantial fraction of human cortical capacity is devoted to social cognition, and many of the brain's most distinctive features, including its large prefrontal cortex and its capacity for language, may have evolved in the context of the demands of social life. The social brain begins its development very early, long before birth, and continues to be shaped by experience throughout life. The developmental trajectory of the brain, from its earliest beginnings through adolescence and into adulthood, is where we are going next. The brain does not arrive fully formed. It is built slowly, precisely, over decades, through a continuous interplay between genetic instruction and environmental experience. The brain at birth is not a miniature version of the adult brain. It is a very different organ, and understanding how it develops is essential to understanding what the adult brain is and how it became that way. Development begins remarkably early. Approximately 16 days after conception, the embryonic cells that will become the brain begin to differentiate from surrounding tissue. A flat sheet of cells folds into the neural tube, the precursor of the brain and spinal cord. By the end of the first trimester, the basic anatomical divisions of the brain are already in place. The rate of neural production during peak prenatal development is astonishing. At its maximum, the brain is generating approximately 250,000 new neurons per minute. By birth, the brain contains most of the neurons it will ever have. But the connections between those neurons, the synapses that give the brain its functional organization, are still largely unmade. The newborn brain is, in an important sense, unfinished. It is wired for the essential functions of life: breathing, feeding, responding to sensory stimuli, seeking the proximity of caregivers. But the higher cortical circuits that will eventually support language, reasoning, and self-regulation are not yet developed. The first years of life are characterized by what is sometimes called exuberant synaptogenesis, a period of explosive synaptic connection formation. The brain of a young child has more synaptic connections than the adult brain will ever have. This overproduction is not a mistake. It is a strategy. By producing a surplus of connections, the brain creates a rich raw material from which experience can sculpt the adult brain, selectively strengthening the connections that are used and eliminating those that are not. The brain reaches approximately 90% of its adult size by age 6, remarkably early. But size is not function. Many of the most important developmental processes that shape the adult brain continue well beyond the size milestones of early childhood. Synaptic pruning, the selective elimination of unused synaptic connections, is particularly active during adolescence. Microglia, the brain's resident immune cells, identify synaptic connections that have not been recently used and eliminate them, a neural expression of use it or lose it. The brain that emerges from adolescence has fewer synaptic connections than the brain of a young child, but the connections that remain are more efficiently organized and more precisely tuned by experience. The prefrontal cortex, the region most associated with planning, decision-making, impulse control, and the weighing of long-term consequences, is the last part of the brain to reach full maturity. Myelination of the prefrontal cortex and its connections with other brain regions continues into the mid-20s. The adolescent brain, though substantial in its capabilities, is operating with a prefrontal cortex that is still in the process of being completed. This developmental timeline has consequences that are both understandable and well documented. The adolescent brain has a highly active limbic reward system, sensitive to the dopamine signals of novelty, social approval, and exciting experiences, combined with a prefrontal regulatory system that is still maturing. The balance between reward-seeking and impulse regulation shifts decisively toward the reward-seeking end during adolescence, before the regulatory capacities catch up in early adulthood. This is not a design flaw. It is a developmental stage, one that may serve the biological purpose of motivating exploration, social engagement, and the expansion of independence that adolescence biologically requires. But it does explain why the risk-taking and novelty-seeking that characterize adolescent behavior have a neural basis, and why the capacity for calm, long-term planning improves so markedly between the late teens and the mid-20s. Adult neurogenesis, the production of new neurons in the adult brain, occurs in the dentate gyrus of the hippocampus throughout life. The existence of adult neurogenesis in humans was definitively demonstrated in 1998 by Peter Eriksson, Fred Gage, and their colleagues, overturning the long-held belief that adult humans cannot produce new neurons. The rate of adult neurogenesis is influenced by experience, aerobic exercise, learning, and environmental enrichment promoted. Chronic stress and social isolation suppress it. The adult brain retains genuine plasticity throughout life, even as some aspects of learning change with age. The speed of new learning may slow in some domains, but the capacity for structural change, for synaptic strengthening, for the growth of new connections, for the reorganization of cortical maps, persists. An older adult who takes up a new instrument, learns a new language, or engages with genuinely new challenges is providing the conditions for neural change. The aging brain shows some decline in specific capacities, processing speed, working memory, the ease of forming new episodic memories, while other capacities remain stable or even improve. Vocabulary typically remains strong. The ability to integrate large amounts of information and draw on accumulated experience, what researchers sometimes call crystallized intelligence, tends to be maintained and in some respects enhanced. The brain's developmental story is one of continuous change across the entire lifespan, from the first weeks of embryonic development to the last years of old age. It is a story of ongoing dialogue between what the genes have written and what experience has edited. And at every stage of this story, some of the most fundamental questions about the brain remain unanswered. Those questions are where we are going next. For all that neuroscience has discovered over the past century, the neuron doctrine, the mapping of brain regions, the identification of neurotransmitters, the discovery of neuroplasticity, the revelation of the default mode network, the understanding of sleep's functions, there remain questions that neuroscience has not yet answered. Some of these questions are at the frontier of current research, likely to yield to sustained effort. Others sit deeper, at the boundary between neuroscience and philosophy, where the usual tools of empirical science may need to be supplemented by new frameworks that do not yet exist. The deepest of these is the hard problem of consciousness. The question is deceptively simple. Why does neural activity produce subjective experience? Why is there something it is like to be you, having your experiences? A complete functional account of the brain, explaining exactly how it processes information, generates outputs, and produces behavior, would still leave unanswered the question of why any of this is accompanied by experience. Why is the processing not happening in the dark, as information computation without any accompanying inner life? This question was articulated by the philosopher David Chalmers in 1995, who distinguished it from what he called the easy problems of consciousness, explaining attention, perception, report, and behavior, which, difficult as they are in principle solvable by neuroscience. The hard problem, explaining why and how subjective experience arises from physical processes, is something else. No current theory adequately answers it, and it is possible that answering it will require not just new data, but new conceptual frameworks, ways of thinking about mind and matter that do not yet exist. Francis Crick, the co-discoverer of DNA structure, spent the last decades of his life working on the problem of consciousness with neuroscientist Christoph Koch. Together, they proposed the claustrum, a thin sheet of neurons beneath the insular cortex, as a possible integrating center that might be responsible for binding disparate neural processes into unified conscious experience. The evidence is suggestive, but not conclusive. The mystery remains open. The function of dreams remains equally unresolved. Dreams occur primarily during REM sleep, when the brain is as active as during waking. They typically involve vivid, immersive experiences, sometimes coherent narratives, sometimes fragmentary and strange. Multiple proposals have been made about their function: emotional memory processing, threat simulation, the random activation of memory traces, generating narrative experience, the integration of new learning with existing knowledge. No single theory has proven sufficient to explain all of dreaming's features, and the question of why the brain generates these elaborate inner experiences during sleep, whether they serve a specific function or are a byproduct of other sleep processes, remains genuinely open. General anesthesia provides another mystery. When a patient is placed under general anesthesia before surgery, they lose consciousness, not only the ability to respond and communicate, but subjective experience itself. When they wake, there is no memory of the intervening time, no experience of any kind. Different anesthetic agents achieve this through different molecular mechanisms. Some enhance GABA activity, some block glutamate receptors, some interfere with sodium channels. But why these molecular changes produce the loss of consciousness, rather than simply sedation or immobility, is not fully understood. Anesthesiologists use general anesthetics safely and routinely, but the mechanism by which they abolish consciousness remains incompletely explained. The precise mechanism of memory storage, the physical encoding of a specific memory in neural tissue, is another frontier. Long-term potentiation, the strengthening of synaptic connections, is a strong candidate for the cellular mechanism. and there is extensive evidence that LTP underlies learning and memory. But the complete picture of how a specific memory is represented in a specific pattern of synaptic connections across a distributed neural network, the engram, is not yet understood. Researchers have been able to identify neurons that participate in specific memories, and in some cases, to interfere with or even plant false memories in animals. But the full mechanistic account of how an experience becomes a memory in physical neural tissue remains incomplete. The gut-brain axis is an area of rapidly growing research that is revealing connections that were not anticipated. The gut contains approximately 500 million neurons, forming the enteric nervous system, and communicates with the brain bidirectionally through the vagus nerve and through the bloodstream. The gut microbiome, the community of microorganisms living in the digestive system, influences brain function and behavior through multiple pathways, including the production of neurotransmitter precursors and the modulation of immune function. Research is finding associations between gut microbiome composition and mental health conditions, including depression and anxiety. The mechanisms are being actively investigated, and the field is young. The full scope of the gut-brain connection is still being mapped. At the frontier of neurotechnology, brain-computer interfaces are progressing from laboratory demonstrations to clinical applications. Systems that allow paralyzed individuals to control prosthetic limbs or computer cursors through the recorded activity of motor neurons are now in clinical trials. The ethical, practical, and scientific questions raised by the direct interface of technology with the brain are extensive and largely unresolved. Optogenetics, the technique of using genetically modified light-sensitive proteins to control specific neurons with light, has transformed the study of causal relationships between neural activity and behavior. By switching specific neurons on or off with millisecond precision, researchers can identify which neurons are responsible for which behaviors in ways that earlier methods could not achieve. The insights emerging from optogenetics are reshaping understanding of circuits underlying memory, fear, reward, and social behavior. Research using psychedelics, psilocybin, ketamine, MDMA, is producing novel insights into consciousness and revealing potential treatments for depression, post-traumatic stress, and addiction. Psilocybin produces marked changes in default mode network activity and in the subjective sense of self, providing both clinical benefits in some cases and a new window into the neural basis of self-referential processing and consciousness. The neuroscience of psychedelic experience is in its early stages, and much remains to be understood. The brain remains, after a century of intensive scientific investigation, enormously and fascinatingly mysterious, which brings us, at last, to where we began, and to the quiet wonder of resting with what we know and what we do not. That is where we are going next, and where we will stay. There is nothing left to explain. The history has been traced, from ancient Egypt's dismissal of the brain as unimportant to the modern era of neuroimaging, optogenetics, and brain-computer interfaces. The geography has been toured: the folded cortex with its 4 lobes, the limbic system with its hippocampus and amygdala, the cerebellum coordinating movement, the brainstem sustaining life. The electrical language of neurons has been described. The action potential, the synapse, the extraordinary density of connection. The chemical language of neurotransmitters has been described. Glutamate and GABA, dopamine and serotonin, oxytocin and cortisol, and the delicate balance among them that shapes every mood and motivation. Neuroplasticity has been described. The brain's lifelong capacity to reorganize itself in response to experience, overturning the belief that the adult brain is fixed. Memory has been described. Distributed, reconstructive, consolidated during sleep, amplified by emotion, shaped by forgetting as much as by remembering. Sleep has been described. Intense biological activity, glymphatic clearance, hippocampal replay, the stages of slow wave and REM, the circadian clock in the hypothalamus. Emotion has been described, woven through all cognition, not opposed to reason, but part of it. Perception and consciousness have been described. The brain as a prediction machine, constructing a model of reality, and the hard problem of why that construction is accompanied by experience. The resting brain and its default mode have been described. Language and music and the social brain have been described. Development from conception through adolescence and across the lifespan has been described. And the mysteries that remain have been acknowledged. Honestly, without resolution, because resolution is not yet available. All of that is there now, somewhere in the brain that has received it, held in whatever form memory takes, distributed across networks, gradually being consolidated, perhaps being replayed even now in the neural activity of a brain beginning its transition towards sleep. What remains is simply to rest in it. Consider the extraordinary fact at the center of everything described tonight: the brain contemplating itself. The human brain, one of the most complex known structures in the universe, capable of directing its own attention toward its own workings, generating curiosity about its own nature, following a description of its own mechanisms with something that feels, from the inside, like understanding and wonder. No other known structure in the universe does this. The brain is the universe's instrument for becoming aware of itself, for the cosmos to look inward as well as outward, to ask questions not only about the stars and the galaxies, but about the very processes that make asking possible. Every thought you have ever had has been a physical event in neural tissue. Every memory you carry is a pattern of synaptic weights distributed across neural networks, a physical record of what was experienced and what the brain decided to keep. Every feeling, the warmth of recognition when something familiar appears, the quiet satisfaction of understanding, the gentle pull towards sleep, has been produced by specific neurotransmitters, acting on specific receptors in specific circuits. And yet none of this reduces the richness of the experience. Knowing that music releases dopamine does not diminish the experience of music. Knowing that love involves reward circuits and oxytocin does not diminish the experience of love. Knowing that consciousness arises from neural activity does not explain away the extraordinary fact of consciousness. The hard problem remains. The explanation of the mechanism is not the same as the reduction of the experience. The pattern of neural connections in your brain right now, the particular configuration of 86 billion neurons connected by 100 trillion synapses, is unique in the history of the universe. It has never existed before and will never exist again in exactly this form. It has been shaped by everything you have ever experienced, every skill you have ever learned, every person you have ever known, every moment of attention you have ever directed. It is, in the most literal and physical sense, you. Right now, the brain is beginning its transition. The frontal lobe is releasing its grip on planning and deliberation. The default mode network is taking precedence. The networks of inner life, of self-reflection and memory and imagination, becoming more active as external demands recede. Alpha waves are emerging, or have already emerged, in the electrical activity of the cortex. The rhythm is slowing. The suprachiasmatic nucleus has been tracking the fading of the day's light. Melatonin has been accumulating, signaling to every system in the body that the time for rest is approaching. The glymphatic system is preparing to begin its work, the channels through which cerebrospinal fluid will flow more freely, clearing the metabolic waste of the day's neural activity. The hippocampus will soon begin its replay, reviewing the day's experiences, consolidating what matters, making connections between what was learned today and what was already known. Some of tonight's journey through the brain may become part of that consolidation. A fragment here, an image there, a connection made between something familiar and something newly understood. The brain that began listening to this tonight is not identical to the brain that is ending it. Every experience changes the brain, however slightly, strengthening certain synaptic connections, creating the conditions for new ones. The journey through these 15 sections has been, in the most literal biological sense, a small but real reshaping of neural structure. The brain is different, however imperceptibly, from what it was when it began. The 86 billion neurons, the 100 trillion synapses, the extraordinary, still mysterious capacity for consciousness and memory and language and emotion and sleep and dreaming and love and loss and curiosity. All of it here, in the quiet of this moment, doing what it does. Sleep well. The brain will take care of the rest.